In this video, we are going to continue the renal physiology portion. So today we are going to learn about the control of extracellular fluid osmolarity and sodium concentration. Normally, sodium ions and associated anions, primarily bicarbonate and chloride, represent about 94% of the extracellular osmoles, with glucose and urea contributing about 3 to 5% of the total osmoles. However, because urea easily permeates most cell membranes, it exerts little effective osmotic pressure under steady state conditions. Therefore, the sodium ions in the extracellular fluid and associated anions is the principal determinants of sodium fluid movement across the cell membrane. Consequently, we can discuss the control of osmolarity and control of sodium ion concentration in the same time. So, although multiple mechanisms control the amount of sodium and water excretion by the kidneys, two primary systems are especially involved in regulating the concentration of sodium and osmolarity of extracellular fluid: the osmoreceptor ADH system and thirst mechanism. So, osmoreceptor ADH feedback system. Basic components of the osmoregulator ADH feedback system for control of extracellular fluid sodium concentration and osmolarity is given. When osmolarity, like plasma sodium concentration, increases above normal because of water deficit, for example, this feedback system operates as follows. An increase in extracellular fluid osmolarity, with practical terms, cause the special nerve cells called osmoreceptor cells located in the anterior hypothalamus near the supraoptic nuclei to shrink. Shrinkage of the osmoreceptor cells cause them to fire, sending nerve signals to additional nerve cells in the supraoptic nuclei, which then relay the signals down the stalk of the pituitary gland to the posterior pituitary. And this action potential conducted to the posterior pituitary stimulate the release of ADH, which is stored in secretory granules or vesicles of ADH, which is stored in secretory granules or vesicles in the nerve endings. ADH enters the bloodstream and is transported to the kidneys, where it increases the water permeability of late distal tubules, cortical collecting tubules, and medullary collecting ducts. The increased water permeability in the distal nephron segments cause increased water reabsorption and excretion of a small volume of concentrated urine. Thus, water is conserved in the body while sodium and other solutes continue to be excreted in the urine. This causes dilution of solutes in the extracellular fluid, thereby correcting the initial excessive concentrated extracellular fluid. The opposite sequence of events occur when the extracellular fluid becomes too dilute, like hypoosmotic. For example, with excess water ingestion and a decrease in extracellular fluid osmolarity, less ADH is formed. The renal tubules decreases their permeability for water, less water is reabsorbed, and a large volume of dilute urine is formed. This in turn concentrates the body fluids and returns plasma osmolarity toward normal. Then ADH stimulates in supraoptic and paraventricular nuclei of the hypothalamus and ADH release from the posterior pituitary. So, neuroanatomy of hypothalamus and the pituitary gland is very important, where ADH is synthesized and released. The hypothalamus contains two types of magnocellular large nervous systems that synthesize ADH in the supraoptic and paraventricular nuclei of the hypothalamus, about 5/6 in the supraoptic nuclei and about 1/6 in the paraventricular nuclei. Both of these nuclei have axonal extension to the posterior pituitary. Once ADH is synthesized, it is transported down the axons of the neurons to their tips, terminating in the posterior pituitary gland. When the supraoptic and paraventricular nuclei are stimulated by increasing osmolarity or other factors, nerve impulses pass down these nerve endings, changing their numbers, membrane permeability, and increasing calcium entry. ADH stored in the secondary granules, also called vesicles of the nerve endings, is released in. Regulated to increase calcium entry. The released ADH is then carried away in the capillary blood of the posterior pituitary into the systemic circulation. Secretion of ADH in response to an osmotic stimulus is rapid, so plasma ADH level can increase several fold within minutes, thereby providing a rapid means for altering renal excretion of water. A second neuronal area important in controlling osmolarity and ADH secretion is located along the anterior-ventral region of the third ventricle called the AV3V region. At the upper part of this region is a structure called the suprafrontal organ, and at the inferior part is another structure called the organum vasculosum of the lamina terminalis. Between these two organs is a medium preoptic nucleus which has multiple nerve connections with the two organs as well as with the supraoptic nuclei and the blood pressure control centers in the middle of the brain. Lesions of the AV3V region cause multiple deficits in the control of ADH secretion, thirst, sodium appetite, and blood pressure. Electrical stimulation of this region or stimulation by angiotensin II can increase ADH secretion, thirst, and sodium appetite. In the vicinity of the AV3V region and the supraosmotic nuclei are neuronal cells that are excited by small increase in extracellular fluid osmolarity. Hence, the term osmoreceptors has been used to describe these neurons. These cells nerve signals to the supraosmotic nuclei to control their firing and secretion of ADH. It is also likely that they induce thirst in response to increased extracellular fluid osmolarity. So both the suprafrontal organ and the osmanum vasculosum of the lamina terminalis have vascular supplies that lack the typical blood-brain barrier that impedes the diffusion of most ions from the blood into the brain tissue. This characteristic makes it impossible for ions and other solutes to cross between the blood and local interstitial fluid in this region. As a result, the osmoreceptors rapidly respond to changes in osmolarity of the extracellular fluid, excreting powerful control over the secretion of ADH and over thirst. So, next topic, stimulation of ADH release by decreased arterial pressure and or decreased blood volume. ADH release is also controlled by cardiovascular reflexes that respond to decrease in blood pressure and or blood volume, including arterial baroreceptor reflexes and the cardiopulmonary reflexes, both of which are discussed. And this renal reflex pathway originates in high pressure regions of the circulation such as the aortic arch and carotid sinus, and in the low pressure regions, especially in the cardiac atria. Afferent stimuli are carried by the vagus and gasopharyngeal nerve with synapses in the nuclei of the tractus solitarius. Projections from this nuclei relay signals to the hypothalamic nuclei that control ADH synthesis and secretion. This, in addition to increased osmolarity, two other stimuli increase ADH secretion like decreased arterial pressure and increased blood volume. Whenever blood pressure and blood volume are reduced, such as that occurs in hemorrhage, increased ADH secretion causes increased fluid reabsorption by the kidneys, helping to restore blood pressure and blood volume toward normal. Then, quantitative importance of osmolarity and cardiovascular reflexes in stimulating ADH secretion. Decrease in effective blood volume or an increase in extracellular fluid osmolarity stimulates ADH secretion. However, ADH is considerably more sensitive to small changes in osmolarity than to similar percentage changes in blood volume. For example, a change in plasma osmolarity of only 1% is sufficient to increase ADH levels. By contrast, after blood loss, plasma ADH levels do not change appreciably until blood volume is reduced by about 10%. With further decrease in blood volume, ADH levels rapidly increase. Thus, with severe decrease in blood volume, the cardiovascular reflex play a major role in stimulating ADH secretion. The usual day-to-day regulation of ADH secretion during simple dehydration is affected mainly by changes in plasma osmolarity. Decreased blood volume, however, greatly enhances the ADH response to increased osmolarity. Decrease in effective blood volume or an increase in extracellular fluid osmolarity stimulate ADH secretion. However, ADH is considerably more sensitive to small changes in osmolarity than to similar percentage changes in blood volume. For example, a change in plasma osmolarity of only 1% is sufficient to increase ADH levels. By contrast, after blood loss, plasma ADH levels do not change appreciably until blood volume is reduced by about 10%. With further decrease in blood volume, ADH levels rapidly increase. Thus, with several decrease in blood volume, the cardiovascular reflexes play a major role in stimulating ADH secretion. The usual day-to-day regulation of ADH secretion during simple dehydration is affected mainly by changes in plasma osmolarity. Decreased blood volume, however, greatly enhances the ADH response to increased osmolarity. Other stimuli for ADH secretion. ADH secretion can also be increased or decreased by other stimuli to the central nervous system as well as by various drugs and hormones. For example, nausea is a potent stimulator for ADH release, which may increase to as much as 100 times normal after vomiting. Also, drugs such as nicotine and morphine stimulate ADH release, whereas some drugs such as alcohol inhibit ADH release. The marked diuresis that occurs after ingestion of alcohol is due to a part of inhibition of ADH release. Then, importance of thirst in controlling extracellular fluid osmolarity and sodium concentration. The kidneys minimize the fluid loss during water deficits through the osmoreceptor ADH feedback system. Adequate fluid intake, however, is necessary to counterbalance whatever fluid loss does occur through the sweating and breathing and through the gastrointestinal tract. Fluid intake is regulated by the thirst mechanism, which together with the osmoreceptor ADH mechanism maintains precise control of extracellular fluid osmolarity and sodium concentration. Many of the same factors that stimulate ADH secretion also increase thirst, which is defined by the conscious desire for water. Central nervous system centers and thirst. Same area along the anterior ventral wall of the third ventricle that promote ADH release also stimulate thirst. Located anterolaterally in the preoptic nucleus is another small area that when stimulated electrically causes immediate drinking that continues as long as the stimulation lasts. All these areas together are called the thirst centers. The neurons of the thirst center respond to injections of hypertonic salt solutions by stimulating drinking behavior. These cells almost certainly function as osmoreceptors to activate the thirst mechanism in the same way that the osmoreceptors stimulate ADH release. Increased osmolarity of the cerebrospinal fluid in the thirst ventricle has essentially the same effect to promote drinking. It is likely that the organum vasculosum of the lamina terminalis, which lies immediately beneath the ventricular surface at the inferior end of the AV3V region, is immediately involved in mediating this response. Then, stimuli for thirst. One of the most important is increased extracellular fluid osmolarity, which causes intracellular dehydration in the thirst centers, thereby stimulating the sensation of thirst. Decrease in extracellular fluid volume and arterial pressure also stimulate thirst by a pathway that is independent of one, stimulated by increased plasma osmolarity, probably related to neural input from cardiopulmonary and systemic arterial baroreceptors in the circulation. A third important stimulus for thirst is angiotensin II. Studies in animals have shown that angiotensin II acts on the subfornical organ and on the organum vasculosum of the lamina terminalis. These regions are outside the blood-brain barrier and peptides such as angiotensin II diffuse into the tissues. Because angiotensin II is also stimulated by factors associated with hypovolemia and low blood pressure. Its effects on thirst helps to restore blood volume and blood pressure towards normal, along with the other actions of angiotensin II on the kidneys to decrease fluid excretion. Dryness of the mouth and mucous membranes of the esophagus can elicit the sensation of thirst. As a result, a thirsty person may receive relief from thirst almost immediately after drinking water, even though the water has not been absorbed from the gastrointestinal tract and has not had an effect on extracellular fluid osmolarity. Gastrointestinal and pharyngeal stimuli influence thirst. In animals that have an esophageal opening to the exterior so that water is never absorbed into the blood, partial relief of thirst occurs after drinking, although the relief is only temporary. Also, gastrointestinal distension can partially alleviate thirst. For instance, simple inflation of balloon in the stomach can relieve thirst. However, relief of thirst sensations through gastrointestinal pharyngeal mechanism is short-lived, and the desire to drink is completely satisfied only when plasma osmolarity and or blood volume returns to normal. The ability of an animals or humans to meet a fluid intake is important because it prevents overhydration. After a person drinks water, 30 to 60 minutes may be required for the water to be reabsorbed and distributed throughout the body. If the sensation is not temporarily relieved after drinking water, the person would continue to drink more and more, eventually leading to overhydration and excess dilution of body fluids. Experimental studies have repeatedly shown that animals drink almost exactly the amount necessary to return plasma osmolarity and volume to normal. And integrated response of osmoreceptor, ADH, and thirst mechanisms in controlling extracellular fluid osmolarity and sodium concentration. In a healthy person, the osmoreceptor, ADH, and thirst mechanisms work in parallel to precisely regulate extracellular fluid osmolarity and sodium concentration. Despite the constant changes of dehydration, even with additional challenges such as high salt intake, these feedback systems are able to keep plasma osmolarity reasonably constant. Increase in sodium intake to as high as 6 times normal has only a small effect on plasma sodium concentration as long as ADH and thirst mechanisms are both functioning normally. When either the ADH or the thirst mechanism fails, the other ordinarily can still control extracellular osmolarity and sodium concentration with reasonable effectiveness as long as there is enough fluid intake to balance the daily obligatory urine volume and water losses caused by respiration, sweating, or gastrointestinal losses. However, if both the ADH and thirst mechanisms fail simultaneously, plasma sodium concentration and osmolarity are poorly controlled. Thus, when sodium intake is increased after blocking the total ADH thirst system, relatively large changes in plasma sodium concentration occur. In the absence of ADH thirst mechanisms, no other feedback mechanism is capable of adequately regulating plasma sodium concentration and osmolarity. So, role of angiotensin II and aldosterone in controlling extracellular fluid osmolarity and sodium concentration. Both angiotensin II and aldosterone play an important role in regulating sodium reabsorption by the renal tubules. When sodium intake is low, increased levels of this hormone stimulate sodium reabsorption by the kidneys and therefore prevent large sodium losses, even though sodium intake may be reduced to as low as 10% of normal. Conversely, with high sodium intake, decreased formation of this hormone permits the kidneys to excrete large amounts of sodium. Because of the importance of angiotensin II and aldosterone in regulating sodium excretion by the kidneys, one might distantly infer that they also play an important role in regulating extracellular fluid sodium concentration. Although these hormones increase the amount of sodium in the extracellular fluid, they also increase the extracellular fluid volume by increasing reabsorption of water along with the sodium. Therefore, angiotensin II and aldosterone have little effect on sodium concentration except under extreme conditions. The relative unimportance of aldosterone in regulating extracellular fluid sodium concentration. Next topic, integrated response of osmoreceptor, ADH, and thirst mechanisms in controlling extracellular fluid osmolarity and sodium concentration. In a healthy person, the osmoreceptor, ADH, and thirst mechanisms work in parallel to precisely regulate extracellular fluid osmolarity and sodium concentration despite the constant challenges of dehydration. Even with additional changes and challenges such as high salt intake, these feedback systems are able to keep plasma osmolarity reasonably constant. And increase in sodium intake to as high as 6 times normal has only a small effect on plasma sodium concentration as long as the ADH and thirst mechanisms are both functioning normally. When either the ADH or the thirst mechanism fails, the other ordinarily can still control extracellular osmolarity and sodium concentration with reasonable effectiveness, as long as there is enough fluid intake to balance the daily obligatory urine volume and water losses caused by respiration, sweating, or gastrointestinal losses. However, if both the ADH and thirst mechanisms fail simultaneously, plasma sodium concentration and osmolarity are poorly controlled. This, when sodium intake is increased after blocking the total ADH thirst system, relatively large changes in plasma sodium concentration occur. In the absence of ADH thirst mechanisms, no other feedback mechanism is capable of adequately regulating plasma sodium concentration and osmolarity. Role of angiotensin II and aldosterone in controlling extracellular fluid osmolarity and sodium concentration. The relative unimportance of aldosterone in regulating extracellular fluid sodium concentration is very important under normal conditions and after the aldosterone feedback system is blocked by removing the adrenal gland and infusing animals with aldosterone at a constant rate so that plasma levels could not increase or decrease. Note that when sodium intake was increased sixfold, plasma concentration changed only about 1 to 2% in either case. This finding indicates that even without a functional aldosterone feedback system, plasma sodium concentration can be well regulated. The same type of experiment has been conducted after blocking angiotensin II formation with the same result. There are two primary reasons why changes in angiotensin II and aldosterone do not have a major effect on plasma sodium concentration. First, as discussed earlier, angiotensin II and aldosterone increase both sodium and water reabsorption by this renal tubule, leading to increase in extracellular fluid volume and sodium quantity, but little changes in sodium concentration. Second, as long as ADH thirst mechanism is functional, any tendency toward increased plasma sodium concentration is compensated for by increased water intake or increased plasma ADH secretion, which tends to dilute the extracellular fluid back toward normal. The ADH thirst system far overshadows the angiotensin II and aldosterone systems for regulating sodium concentration under normal conditions. Even in patients with primary aldosteronism who have extremely high levels of aldosterone, the plasma sodium concentration usually increases only about There are two primary reasons why changes in angiotensin II and aldosterone do not have a major effect on plasma sodium concentration. Angiotensin II and aldosterone increase both sodium and water reabsorption by the renal tubule, leading to increase in extracellular fluid volume and sodium quantity, but little changes in sodium concentration. Second, as long as the ADH thirst mechanism is functional, any tendency toward increased plasma sodium concentration is compensated for by increased water intake or increased plasma ADH secretion, which leads to which tend to dilute the extracellular fluid back toward normal. The ADH thirst system far overshadows the angiotensin II and aldosterone systems for regulating sodium concentration under normal conditions. Even in patients with primary aldosteronism who have extremely high levels of aldosterone, the plasma sodium concentration usually increases only about 3 to 5 meq per liter above normal. Under extreme conditions caused by complete loss of aldosterone secretion as a result of adrenalectomy, or in patients with Addison's disease, severely impaired secretion or total lack of aldosterone, there is tremendous loss of sodium by the kidneys, which can lead to reductions in plasma sodium concentrations. One of the reasons for this is that large losses of sodium eventually cause a severe volume depletion and decreased blood pressure, which can activate this thirst mechanism through the cardiovascular reflexes. This activation leads to a further dilution of the plasma sodium concentration, even though the increased water intake helps to minimize the decrease in sodium fluid body under this volume under these conditions. Thus, extreme situations exist in which plasma sodium concentration may change significantly even with a functional ADH thirst mechanism. Even so, the ADH thirst mechanism by far the most powerful feedback system in the body for controlling extracellular fluid osmolarity and sodium concentration. Next, salt appetite mechanism for controlling extracellular fluid, sodium concentration and volume. So, maintenance of normal extracellular fluid volume and sodium concentration requires a balance between sodium excretion and sodium intake. In modern civilization, sodium intake is almost always greater than necessary for homeostasis. In fact, the average sodium intake for persons in industrialized cultures who eat processed foods usually ranges between 100 and 200 m equivalents per day, even though humans can survive and function normally while ingesting only 10 to 20 m equivalents per day. Thus, most people eat far more sodium than it's necessary for homeostasis, and evidence indicate that our usual high sodium intake may contribute to cardiovascular disorders such as hypertension. Salt appetite is due in part of the fact that animals and humans like salt and eat it regardless of whether they are salt deficient. Salt appetite also has a regulatory component in which there is a behavioral drive to obtain salt when a sodium deficiency exists in the body. This behavior drive is particularly important in herbivores, which naturally eat a low sodium diet. But salt craving may also be important in humans who have extreme deficiency of sodium, such as that occur in Addison's disease. In this instance, there is a deficiency of aldosterone secretion, which causes excessive loss of sodium in the urine and leads to decreased extracellular fluid volume and decreased sodium concentration. Both of these changes elicit the desires for salt. In general, the primary stimuli that increase salt appetite are those associated with sodium deficits and increased blood volume or decreased blood pressure associated with circulatory insufficiency. The neuronal mechanism for salt appetite is analogous to that of the thirst mechanism. Some of the same neuronal centers in the AV3V region of the brain seem to be involved because lesions in this region frequently affect both thirst and salt appetite simultaneously in animals. Also, circulatory reflexes elicited by low blood pressure or decreased blood volume affect both thirst and water appetite at the same time.